Good. Hello. We were here, I think, about 3 years ago. We came up with Max 4 again at the time. We had a smaller conversation with Astra Zeneca. I'm pretty sure it was in about 2013. I might be a year or so out. It's a pleasure to be here. Thank you to Anna, who's disappeared behind there. Where's she? Anna's disappeared. There, hiding there. Thank you very much to Anna for all the organization. I'm Ed Mitchell. I'm head of biz dev at the SRF down in Grenoble in France. I also get to do a little bit of science still in my spare time, where I do a little bit of malaria and HIV research with collaborators at Kiel, the ILL, and over at Wits University in South Africa. Synchrotrons have been around for some time. You go back to 1945. This is the first cyclotron at the time, built by General Electric just after the war over in the US. A little beast, made out of glass, about this big, circulating electrons, and they saw synchrotron light for the first time. It was only because they had a fluorescent piece of paper or card on the side of the lab, and by accident they could see the light coming out from the electrons being circulated. That's a low energy machine, so the light coming out was UV, and it was making the little piece of paper fluoresce. These days, there's loads of synchrotrons now. These are the main European ones. Most of the countries in Europe have a facility or have access to the SRF over in Grenoble. These are now 1 kilometer sized machines, high energy. The SRF, we're at 6 GeV. I think max is 3.0 GeV. So the two are complementary, delivering different types of X-rays. How do they work? Can we try the video, please? See if it works. This is the SRF, but they're all the same, right? It's a big ring. Inside, there's a high vacuum ring where we circulate electrons at very close to the speed of light, 0.9999999 times the speed of light. They rip through magnets called undulators, high magnetic fields, which make the electrons ripple. They undulate, and with every undulation, they emit a packet of X-rays that we harvest along beam lines, focus, tune them, and then use them for different types of experiment. At the SRF, we've got 43 different instruments, and I think at Max Lab, probably targeting about a similar number once Max Lab is fully built in a few years' time. The SRF is international. We have 21 different countries involved in funding the SRF. I'm up here in Scandinavia. Here's NordSync, which is a consortium of Denmark, Finland, Norway, and Sweden, contributing to the SRF. Our budget's about 100 million euros, about just over 600 staff. And we're set up actually as a private civil company under French law. Just some key figures down here. We're based right just on the edge of the French Alps. Nearest ski resort for anybody who's coming down to visit is about 30 minutes away. A good time to come down is kind of March, if you were thinking of coming down. We have about 6,500 users coming to use us every year from around the world. We have about 2,000 academic proposals. Industry is more than welcome to apply for free beam time as well. About 900 are accepted, and when they're accepted, we pay for everything: travel, accommodation, the beam time, laboratories, what have you. We have about 5 publications coming out every day, and just over 25,000 articles have come out in the 20-year period since we started operation in 1994. In fact, we did a survey a little while ago, and about 30% of the work we do at the SRF has got an industrial link. The academic work coming in has got industrial backers, or it's going to be patented or licensed, or it's working with industry in some way. And every year we generate about 2 million euros in industrial commercial income to the SRF through providing services and a bit of tech transfer. And importantly, in-house we can fund about 50 different PhD students, often in collaboration with industry partners or academic partners. We're housed on the Grenoble Innovation Campus, a site with about 10,000 scientists down in Grenoble, going from Grenoble Railway Station, somewhere over here, all the way out to the synchrotron between the two rivers. And this combines 8 different partners in the Innovation Campus, including partners like the French Atomic Energy Authority, a couple of universities, and a management school. So it makes for a very lively campus that we're now trying to work more closely together. On our site, this is our site, the European Photons and Neutrons campus, right at the end of Giant, the synchrotron, big round thing, you can't miss it. This is the Institut Larry Langevin, and Trevor, where's Trevor? Who's at the back over there, is going to say something about neutrons a little bit later on, I think this afternoon. This is our sister institute where they generate neutron beams instead of synchrotron X-ray beams. On our site, we also have the Grenoble outstation of the European Molecular Biology Lab, where they have some amazing crystallization facilities, and they help us a lot with instrumentation developments on the MX beam lines at the SRF. And just moved on to our site is the French Institut de Biologie Structurale, where they have cryo-EM facilities, 900 megahertz NMR. And we hope, I think Gordon might talk about it later on, during next year to have one of the Cryos 10 machines arriving at the ISRAF, which will cooperate between us, EMBL, and IBS. Let's just go backwards a bit in time here, right? Does anybody play golf? There's two people sort of, well, maybe, not too sure. Okay, back in, when's this? 1928. So this is what, about 30 years after X-rays were discovered by Röntgen. This company, US Royal Golf Balls, was using X-rays for quality control for their golf balls. So it's simple X-ray radiography, right? Because if your golf ball, the inside of your golf ball isn't spherical, when these guys are hitting it down the fairway, it's not going to go in a straight line. It's going to go wonky somewhere. Certainly, if I hit it, it does go wonky. That's you've got wonky golf balls. You need some X-ray quality control. Just send us some of your balls, and we'll check them. So what these guys were doing radiography. These are the competitors here, where you can see it's kind of wonky egg shape. It's no good. And they were doing quality control. And they could just look through the golf ball and show that the golf balls that they were making had nice spherical interiors, and they were going to improve your golf score from 102 to 91, so saving 10. It's okay? Okay. X-rays today, right? It's the same thing. We do the same kind of things at the synchrotrons now, but we're doing it faster, smaller, quicker, better. So obviously with synchrotron X-rays, you can look inside objects. You can do that at the hospital as well. But the synchrotron, we can do that at very, very fine scale. So you can look in and zoom in on an area and see it at a very, very fine spatial resolution. So at the moment, our best beamline at the SRF can go down to about 20 to 30 nanometers spatial resolution in tomographic mode, I think. And Elliot is going to talk about that a bit more later on. We can go very fast because we've got lots of photons. Got a huge quantity of photons. So using those, we can either go very fast, looking at things that are dynamically changing, so filming them as they're moving and changing. And we've done work with medical devices, watching the device as it's doing its thing inside whatever object it might be in. We've done some nice work with a European client in that domain. But also because we've got lots of photons, we can also do lots of samples. So suddenly doing things very, very quickly for statistical analysis becomes possible. We're working with a client at the moment who wants to send us 50,000 samples for powder diffraction. And we're trying to see how we can make that possible. We hope to have the first samples going through in November. Using a lab source, it would be logistically not possible to do that. But because we've got lots of photons and the exposure times are quick, well, suddenly we can start doing this kind of work. And synchrotron X-rays, same as neutrons, allow you to look inside real objects under real conditions. So you can put real objects on the beamlines, watch them dissolving, watch them getting heated up, watch them changing in humidity, for example, and scanning objects as they're manufactured. So we've had batteries on the beamlines, looking inside, watching the chemistry as they're being charged and discharged. We've had lithium-ion batteries exploded on the beamlines, which is quite a hot topic at the moment, and trying to understand what the processes are involved in when they fail. So at the ISRAF, these are some of the clients we work with in terms of industry. We don't show a client unless they've given us permission. Otherwise, it's confidential and fully proprietary. There is Astra Zeneca is here somewhere. We've had samples of all these different types and more on the beamlines, but a lot of the mainstay of the work that we do still at the moment is with the pharmaceutical industry, be it for drug discovery or other areas. But we've had frozen peas on the beamlines, batteries I've mentioned, catalyzers, margarine from Unilever, I think it was, aerial washing up liquid from Procter & Gamble, looking at how these objects, looking at their microstructure, looking at the phases, trying to understand the processing conditions that could make better ice cream. It's not a nature front cover, but this was Procter & Gamble, I think. It was Unilever, Unilever with the ice cream. And we were looking at the microstructure, trying to look to see how the processing could make it better. It's not a problem up here because it doesn't get that hot in the summer. But in Grenoble, where we hit maybe 35, you buy your ice cream, it melts a little bit in your car, you take it home, you put it back in the freezer, and the structure changes. So the company wanted to look at the microstructure to see how better to optimize their processing conditions to make the ice cream more stable with time and temperature. Okay. How am I doing for time? It's okay? So down at the ISRAF and at Max 4, the other big facilities, we can cover domains. You're the experts here when you start seeing some of this stuff I'm going to put up. We can work from atoms across the human scale. Both the ISRAF and Max 4 have or are planning large-scale imaging labs and animal facilities. At the atomic scale, and you're going to hear about this from the various speakers during the day, we can work at looking at drug structures. The obvious example is structural biology, protein crystallography, where we look at protein structures. People send us samples, we send back data, and they look at how to model better ligands in there, how to develop new drugs, be it through fragment screening or other lead targets. We can do small molecule work, powders I've mentioned already. There's Sachs, biosachs, and paired distribution function. I think this is all going to be talked about later on. And that's at sort of the atomic, maybe the molecule level. Going a bit higher in scale, you move to more direct imaging. So I think Elodie might talk about imaging of pills later on, watching a pill as it's been dissolving. We can also do X-ray fluorescence, because every element has its own X-ray signature. So you can pick out an element for an object and see how it's distributed. We've done work on implants, looking where certain elements are composed within the implant to understand what's going on. And Diamond, I don't know whether Max is planning. Diamond have a very nice cryo X-ray microscopy facility that can do effectively sort of confocal microscopy at the cellular level, but using X-rays. Going further up, it's basically the same set of techniques, but a larger scale. Looking at implants, we've done work on dental implants, for example, looking at how the fatigue changes with time between the titanium elements of the implant. And you can start looking at phenotyping. So looking at, for example, the root systems of plants to understand how they're changing with different measures that are being put in, be they weed killer or hormonal controls. And at the very highest level, we've done work on medical devices that I've already mentioned. And on the medical beamline at the SRF, we have a medical therapy program that has been looking at how to use stereotactic radiation therapy and microbeam therapy to help with brain cancer and other types of cancers. So we've had people on the beamline, and we have, I think we're at the moment still the only facility in Europe with an animal house, but I think Max 4 is going to be catching up very, very rapidly. How do we work with industry? Just to give you a flavor. Firstly, we do have proprietary services, be they mail-in, complex. This generates about €1.5 million. We actually have about 150 clients at the moment. All the IP belongs to the client, and we base this, I think Max will probably be the same, on cost recovery. So our goal is not to make a profit. Our goal is to get industry using these facilities that are based around Europe. As I mentioned before, industry is more than welcome to apply for free beam time. The next deadline at the ESRF is coming up in March, I think it is. If you publish the results, you can have the access for free. It's a competitive access, but we have examples where industry has come in and got peer-reviewed beam time and is using the beamlines. We're doing more work now on collaboration and grants where we've got industry-sponsored staff, postdocs. I think we may have a postdoc with through AZ coming at some point soon. And we've got some other postdocs, PhD students funded by industry at the moment. And we're active in European grants. NanoElec is a French public-private partnership with €6.5 million for the ESRF and the ILL to learn how to better serve the nano and microelectronics industry, which is very strong in the Grenoble area, where we have ST Microelectronics, Soitec, and Schneider. And we're working directly with them to understand what do they need on our beamlines so they can better serve their industry. And we're actually now building a €1 million instrument specifically for the electronics industry on one of our beamlines. This is an aside. We also have a lot of our own internal technology that we outsource, out-license to other partners and sell directly to. We haven't sold anything to you guys yet, actually, I think. We should change that. But we sell, sorry? Yes, we are. I'm going to mention that later on, the copying part. Right? But we sell a number of devices to the other light sources, and we outsource some of the instrumentation. So I just wanted to show very quickly a couple of examples. Gordon and others, I think, are going to repeat some of this, but I think it was worth saying twice. So some of the work we do is trying to make it easy for industry to use us, because we are seen as a bit of an exotic, esoteric image facility, but we try and make it easy with mail-in services, building things so that you can just use the facilities with great ease. Can you try the video, please? So this has been an evolution of the structural biology beamlines over Gordon, where is he? Gordon over there. Last 20 years or so, building up to a system that is now fully automated. The samples arrive from academia and industry. They're put into the robots. The sample changer works, puts it onto the beamline, calculates the data collection strategy, processes the data, can even try and solve your structure, I think, and it just chugs away routinely, the same thing day in, day out. And that lets us charge. People access this for starting at €70 per sample. And when I arrived at the ESRF 1,000 years ago, this was about €10,000 a sample. But the technology that Gordon and the group have been leading has changed things so radically that this has now come down by 2 orders of magnitude. So really democratizing that kind of access for industry for proprietary work. So even SMEs can afford this kind of access. A more complex collaboration is completely to the other scale. This was some work with Varian, who supply some of the world's radiotherapy units and a lot of other devices. And we did a 2-year project with them, making use of some of the expertise we've got on the ID17 beamline at the SRF, which is the medical beamline, where we have experience and expertise in radio doses. And they wanted to come down and see whether microbeam radiation therapy could be of use for their systems. So they wanted to explore whether having a comb of microbeams of X-rays, in their case in lung cancer therapy, could help. So this is a 2-year program using our expertise. We outsourced some MRI work to the local neurological institute, and we were looking to see whether lungs treated with microbeam radiation therapy did undergo pulmonary fibrosis after treatment. Because that's one of the causes, I understand, I'm a bit outside of my expertise here, is one of the causes of problems in radiotherapy for lungs. And Varian wanted to come down and study and see what was going on. And this is a good collaboration for us. It's using our expertise. It's a proprietary access. But there's also a publication that we expect to come out from it as well, which is good for our scientists and good for Varian as well, that we get an academic publication coming out. And that should be coming out, I hope, very, very soon. We work on training, which is an important aspect of what we do at the ISRAF. We have undergraduates coming down during the summer, working for 2 to 3 months in Grenoble across Giant, doing small-scale research projects, be it with the ISRAF or with EMBL or the CEA or the universities. And at the ISRAF, we have placement students, where we have undergraduates coming down to our site for up to a year. And we've done this so far with AZ, GSK, and Novo, coming down using the facilities at both the ISRAF and the ILL to do joint little research programs. Publishable research usually, but it kind of shows off what we can do and helps introduce the companies to the different techniques that we have at the ISRAF and on our site. As I mentioned, we do have PhD students working with industry. This is one example with Synthelis, which is a local biotechnology company in Grenoble, who have expertise in cell-free protein production. We're working with them, again, jointly between the ISRAF and ILL, and with WITS in South Africa, Kiel in the UK, and the University of Grenoble Alpes, the local Grenoble University. We're working on HIV and infection processes, looking at the structure of certain proteins involved, and working with Synthelis using their technology. So it's a very nice worldwide collaboration with 2 young PhD students, Vinesh, who's originally from Mauritius, and Jennifer from the UK, who was originally a placement student with us with Glaxo. She was so good we got her back. And these are 2 young 2 young kids experiencing, they're not children. That shows my age, right? That's bad, right? I know I'm getting old, but they're not children. They're young researchers, 2 young researchers on our site, experiencing cross-border and cross-disciplinary research linked with industry. Okay, right? They're not children. They are actually very, very good students. This is being videoed as well. Don't show this to Vinesh and Jennifer. Okay, the ISRAF, we are copying Max 4 somewhat. So we started a few years ago with an upgrade program. We've built out some buildings at the ISRAF to make longer beam lines, which allows us to focus the beam down and still have exciting sample environments around it. Because the longer the beam line is, the more space, it's just physics, P over Q for those that did physics, in terms of focusing. So you have more space around the sample to put in, be it pressure, temperature, cryo, whatever kind of environment that you want around your sample. But more importantly, we're taking some of the ideas from the Max 4 magnetic lattice, which is the big storage ring where the electrons are, and we're taking that, changing a little bit, and then reinstalling something at the ISRAF in 2020, which will make our X-ray beams about 2 orders of magnitude better than they are today. This means maybe things like 5 nanometer X-ray beams for imaging, microscopy purposes. It means a 100-fold increase in orders of exposure, speed. The dynamics and the work that we can do using those beams will be amazing. It does mean that we'll be closing down in December 2018 for 18 months, just as Max is, I think, probably largely available. And this is going to be a very exciting period for the ISRAF, and this will make us the most brilliant source in the world in August 2020 when we come back up. A lot of the science drivers for this work needs microscopy. In there is a big one, it's health and structural biology, medicine over here, but there's also new materials, be they for medical devices or for composites for cars or for batteries, or we can start looking at very, very fast biology. Not as fast as the FELS, but fitting in between synchrotrons and what the free electron lasers can do. And this will be coming up with a set of even more beamlines, as I said, in about 2020. So synchrotrons, over the last, the ESRF was the first third generation source becoming available in about 1994. And over the last 22 years, synchrotrons have become really a routine, de facto part of the European science scene as an advanced materials analysis tool. But we're trying to make them available to industry so that you guys can make more use of them. And they've revolutionized basic and applied research and innovation. And now everybody expects to have access to these things. There's a community of, I think, about 30,000 scientists in Europe that count on having access to these tools every day. They're drivers of technology. We build new, better detectors, better vacuum components, other instrumentation that have spillover to other areas. You've seen that we do education with students and young researchers. And at the ESRF, we have, I think, about 60 different nationalities represented in our staff. So we're very much centers of cross-cultural exchange. We are open for commercial exploitation and collaboration with industry. It's not easy for us because it's not easy for us to understand your world. Max 4 has an advantage because Thomas here is ex-Astra Zeneca and has a great insight into what you guys need and what we can do. But we are open and we want to collaborate, and we're here to try and understand more and to talk with you and see what more we can do for both ESRF and Max 4. So thank you for your attention, and I look forward to discussions. Thomas.
Ed was kind to share his presentation ahead of me creating my own, so I've tried to exclude things and overlaid or avoid overlaps. Even though the repetition is the mother of learning, so some things I will repeat from my own angle and my own twist to try to explain the usefulness of these facilities. Yes, I've been on this stage many times before, and I think I recognize half the audience. So in that sense, it feels very comfortable. But now I'm in a completely new environment, a facility that covers all areas of research. So what I'm grasping with is not to understand pharma, but it's actually to understand all other areas of science and how we contribute. So I prepared a little talk, and I think it might be worth saying that we are a national research infrastructure. In fact, it's the most expensive that have ever been built in Sweden. And we are hosted by Lund University, but we're actually funded with money from the Research Council and all the 12 Swedish universities that was available at the time, and actually put their own money into the facility. So we really feel like a bottom-up facility that has the full support. And since then, there's also been universities in Denmark and Finland who's actually contributed directly money into the operation. And that sort of feels good, and not to be a political project, but actually being a research-driven product. I will start by talking about what we are not, because I think there are some misunderstandings about these facilities that tend to survive among politicians, which I've been talking to lots of lately, among newspapers and journalists. So I will try to correct some of those. I will talk about the Max 4 project, and that Max is not new. It has a 30-year history of photon science. Little bit of what our ambition is. I think Ed has done such a great job in explaining what types of science we can support in what ways, so I will not dwell too deep into it. And I will talk a little about what we are doing in terms of trying to connect with industry, even though we are not really operational yet. So what we are not the ESS. ESS is our neighbor, about 1 kilometer away from our site. We're sharing the site down in Lund. ESS is a neutration source to be compared with ILL. And I think you will learn a lot from Trevor later on here, what you can do with it, so we are highly complementary. But as I said, Max 4 is a Swedish product, while ESS is like ESRF, very much a European project, consisting of 70 member states. And what we do, as the fundamental techniques are the same, being used, being diffraction or imaging or whatever, but we use different probes. So we use photons and they use neutrons, and the results coming out is very different and complementary, I should say. So we don't have any competition, we have collaboration. And I should add to what Ed said, this is a very collaborative field. This facility wouldn't exist and be so cheap to run and operate and be as good if there was not a true collaborative spirit between the facilities. And we have copied you, Ed. We have copied your control system, for example, IcePap, etc., etc. So there are lots of examples where we've sort of helped each other out. Another tremendous misunderstanding tends to be that we are not a research facility. We don't have scientists doing all kinds of research. I think ESRF has a postdoc program and some in-house research, but we are mainly a research infrastructure. That means that scientists from all these universities I mentioned actually come to us to do the experiments, and they are the guys doing the science. We do have scientists, but they are mainly involved in developing methods and keeping these experiments that we have, or experimental stations, up to date and keeping advancing so they can do more science and broader science and different science going forward. So this means that we generate a lot of impact on society and on research and innovation, but that research that we help catalyze or produce is actually happening all around us, not just in Sweden, but also in other countries when they come and visit us. Had it not been that these experiments needed these rings and the systems that generate the source, I think they would have probably been spread around the country and been very much closer to the research groups that are the expert users of these facilities. There might have been in Chalmers, KTH, and Umeå. But the fact that you need this brilliant source is the reason they're placed in Lund. So Max 4 is not a Lund thing. It's not only local interest. It's actually something that supports the whole country and has impact throughout the country. So these are the misunderstandings. As I said, there's a long history. We have 30 years of synchrotron research, and the initial research started in a small basement as a subversive effort by some physicists in Lund who created the first accelerators to generate X-ray. And there's been, as indicated by the 9.4, there have been at least 3 accelerators prior to Max 4 housed in this laboratory in central Lund. Lucia last year, we closed it down. So that means while building the new facility, we also kept operating the old one, which was a huge effort by the staff. And we inaugurated midsummer this year with the King and the Prime Minister, the new facility. And that date was chosen because it was the brightest day of the year. It wasn't really meant to indicate that we are fully operational. We are still working, but we actually had produced the first light and the first experiment to that date. So it was time to celebrate. And I should mention that the King has been very involved in this for many, many years, and he's been there on several occasions when projects have started and so forth. But this time we thought we should invite the Prime Minister, but the Prime Minister sits on the money, and money is good. So we invited the Prime Minister and said, yes, thank you. And later we got a telephone call from the people around the King saying, what about this invitation to the inauguration? So the King actually invited himself. So it was inaugurated by Stefan Löfven in the presence of His Majesty the King, which was not bad. X-rays has come a long way, and Ed talked somewhat on what synchrotrons contribute, but actually really the first experiment done by Röntgen on his wife showed the insights you could get by using X-rays. And they have this penetrating ability. That means you can study objects without cutting them apart and see what to look at on the inside. But there's also another reason, a property that well, there's several properties, and I will mention a few of them. One being that if you want to study an object that is really, really small, you need to use some kind of radiation that has the same size, length scale as the object you want to study. And if you want to get to atomic resolution, you need X-rays. Simple as that. I like this experiment for many reasons. He never wrote a paper about it, and he still got awarded the Nobel Prize. And it took 1 year for actually to appear in Vienna, shops that provided this service as a commercial service. So it was great science and it was great innovation. So I think Marlon has. When did the Max 4 really started as a discussion? more than 10 years ago. Yeah. So Max 3 was starting to get aging, and we were well behind ESRF, for example, and many other facilities, and Astra Zeneca, and including myself, has abandoned Max 4 for doing some of the work. So we needed to do something. So there was some smart people who sat down in Lund and tried to think of a new facility and how they could actually afford to build one, because these are very expensive to build. And one of the discussions was that, what you really need. You need, if you possibly can, you want X-rays over such a wide spectrum or energy range as you possibly can. You want high brightness, etc., etc., and low emittance. It means as many photons over defined areas as possible. So I really came to the conclusion this shouldn't really be done by one machine. So in the Max case, we did actually have 3 machines. One for high energy, which is in our case is 3 GeV, not as high as ESRF. But as opposed to ESRF, we also have a soft X-ray or a low energy 1.5 GeV ring. That is very useful for spectroscopy and material science. And we have a linear accelerator from which we can generate short pulsed light to study very fast processes. And fitted together, this is what it looks like. So we have a linear accelerator that starts by accelerating electrons. Some electrons disappear up to low energy rings. After another 150 meters, you are up in 3 GeV, so it feeds the big storage ring. And at the end of it, you have this short pulse facility where we, in combination with lasers, can study a lot of different types of chemistry. So it really should be called Max 4, 5, and 6, but it wasn't. So we're stuck with Max 4. It was expensive to build, and we don't have plans to build 43 beamlines at ESRF. We could not afford that as a national facility, so we're aiming for a number around 26. So what's unique with Max 4? The uniqueness is, as Ed said, the lattice. If you want then electrons to stay circulating in the ring, and you want them to stay circulating in an ordered and very dense fashion, you need to use different type of magnets. Some of the magnets make them stay together because they really want to go apart. Some of them need to steer them so they turn in the circle. And some need to bring back more energy in so they don't lose energy and the energy level of the ring stops. So the better property in the better ring is really about how many magnets you can fit into a certain distance. And that was really the trick that Lund managed to solve differently than anyone else. So what Sweden is good at is machining things out of iron blocks. We have machining companies who can do this with a precision that's just phenomenal. So the concept was to machine actually magnets out of a block. So these cores are the same block of steel as all the other magnets are fitted into, and you then put on these copper coils to create magnets. So it's prefabbed into one block that's very rigid. So everything is pre-aligned with a precision that you cannot achieve if you build this individually, which was the way it was done before. So instead of it being a massive alignment thing, with mechano building, constructing, which took a lot of time and was very expensive, we got these prefabbed in 2 factories, in Danfysik in Denmark and Scanditronics in Vissefjärda in Småland, and delivered to the facility to be fitted together like a Lego instead of a mechano. And this saved cost. It enabled this precision that has never been achieved before. So this was suggested by Mikael Eriksson, who is a professor emeritus now at Lund University. Everybody laughed at him for about 10 years, and this is not possible. The magnet's going to interfere, etc., etc. Nobody's laughing anymore, and he's now traveling around the world as an advisor for all these upgrades that's happening around the world. And in terms of brilliance, we're going to beat, this is logarithmic, so we're going to beat everyone else with quite a bit. This is a Stanford synchrotron that came around online about a year ago, and we're still going to be stronger. APS, which was the big, might not be here on that, which is the big synchrotron in the US, was actually scheduled for an upgrade that was cancelled because they realized even after the upgrade they're not going to be better than us. So they are back to the drawing board and now being funded again for an upgrade. But it's worth pointing out that in certain energy ranges, if you want to look really through thick materials, which is not my life science, but steel, you might be better off going to Hamburg or SRF going forward. So we all have our niches and the different synchrotrons. So these are the planned upgrades and rings that's going to copy us going forward. including new things being built in Beijing and Thailand. So it's been lots of talk about brilliance as we being the most brilliant facility, but there's actually another property that is as important, particularly for some types of experiments, and that's coherence. And today's synchrotrons, the beam is not very coherent or ordered or structured, what you would like to call it, or more laser-like, which means it's almost only possible to study structured materials, and that could be crystals that Tove or someone else is bringing in. While if you have more coherent beam, it becomes much easier to study more disordered samples, and a lot of samples in life science of interest is actually to be characterized as very disordered. So this is important as the brilliance, and MAX4 will have a higher coherence than any other synchrotron going forward. So that's one of the properties we should really make use of when we build experiments. So all these numbers that have been shown past was only calculated numbers, but now we actually validated most of them. So for the hard X-ray, accelerated the 3 VGings, we have all the measurements that show that we will live up to spec once we are up and running at full speed, which is very comforting. People sleep a lot better. This example of a start of an experimental station. So from this circulating electrons, you extract light with these insertion devices, which makes electrons go slalom. And when they go slalom, they get sweaty and they emit light. And that light is funneled out through the ring into an experimental station. So this is where they go and out to the wall again. And normally here, which is missing in this picture, is actually monochromator. You can, this is a monochromator. You can select the wavelength. You tailor basically in this room the beam that you would like to have for your experiment, the wavelength, the size of the beam, etc., etc. Going one step further, you come to the experimental station, and this is what they look at MAX4. And inside, this is equivalent of the protein crystallography beamline that Ed shows, and this is going to be as optimized and hopefully in the end also remotely available for data collection going forward. So you don't build a synchrotron in one day, so beamlines like we have at Max Lab will come online over the next years. We simply don't have the capacity, or you can't build them all at once. So we're going to slowly come online exactly when ESRF shuts down, so that's good timing. And we welcome anyone who we can support with our beamlines from ESRF to come over. A little bit about the past. I think this is pretty amazing. So last year we actually broke the record whilst building the new facility. We had the record number using at the old facilities, about 1,000. And they come from all areas of science. And I think we will expect chemistry and life science to grow in the new facility based on the beamlines we're going to build. And we also in the past worked with a lot of companies, including Astra Zeneca and PHARMA. So we have 14 beamlines, and I will not go through them in detail what they do. A lot of them can do more than one thing. They have more than one end station. So the limitation of what these can do is really, I shouldn't be limited by imagination or what you think, but it can be technically limited in the sense that you might want a very specific sample environment that is going to do your type of experiment. You want to pull apart something at 500 degrees while measuring how it cracks and etc. And that might be that we need to design or work together to define what we call a sample environment that can do your specific type of experiment. But this is the sort of discussion that we would like to have with the user community, exactly what type of experiments you want to do, using some of our techniques, and how can we work together to create the sample environments and support for the experiment to make them work. I might well have this slide up saying that we are, yes, initially we were funded by the universities, but we have countries coming in. Finland and Estonia was one of the first one who financed the beamline. As of late, Denmark has realized there is more than neutrons. Instead of just investing in ESS, they have now invested in a beamline at MAX4. And this was symptomatically not done by the government, but actually by the universities, Aarhus and Copenhagen, and the regions who find synchrotrons useful. Putting a little bit of different touch to why I'm fascinated by synchrotrons and why I think they can be important for life science in particular is that no matter what biology you study, whether it's bones, you really need to understand things in very different length scales to make sense why a bone go brittle or why it can be so flexible, etc. So it's really about having techniques that from where you can study the molecular structure, as Ed said, all the way through the intermediate structures up to where you can see the full bone. But this goes for any. It's the same about muscles. You cannot understand a muscle unless you study it at different length scales. And same goes for the brain. You need to understand everything from ion shells work into neurons and how they're connected and how different areas of the brain connect. And to do that, we have sort of aimed at having beamlines or experimental stations that can support all these different types of length scales. So from atoms all the way through biomolecules into structures like membranes and cells and tissues and so forth, to support the different areas of biology. You could do a similar sketch for material science in general, etc. For any science area, I think it would be true. But this is my ambition to build in support of life science. And there is another dimension, which is the fourth dimension. That's about studying things where it actually move. And this is another advantage of synchrotrons, that you can do very fast measurements. And if you can do fast measurements, you can catch movements, whether it's about bone bending or, in this case, a fly flying. You will hear more about this type of imaging from Raymond, who's arriving around lunchtime today, talking about. But when I saw this movie and how you can study muscles, flight muscles, and lungs, etc., in insects when they are still alive, I got pretty fascinated and sort of realized what that can be done elsewhere. So he will talk more how he's working now on lung models to study the effects of ventilation and ventilation damage. He's studying hearts to look at damage from heart or heart attacks or damaged tissue, etc. So it's a lot of functional studies all of a sudden just becoming available because they improved coherence. You can get the dose down, you can start to look at things with still retaining the biological relevance and not getting the radiation effect in the biology too much. It's a lot of potential. Another advantage of making things at speed is if you really want to trust your experiment, and I think this is very important for companies at least, that you have your statistics to prove that what your conclusion is actually right. So to automate experiments and derive the statistical significance is really important before you make your statements. And I replaced these length scales with different people, so we have lots of colleagues with me today. And they can talk to you more about the different techniques we have and the different length scales. This is Raymond I talked about. Olivier can talk about diffraction, studying, for example, membrane surfaces, but also powder and tablets or whatever. You can use that techniques for many things. Christopher, expert on SACs or solution scattering studies. Johan is involved in the protein crystallography, bed development, and Kaisa knows everything about spectroscopy if you want to know what things are made up of. So make use of these people, and they're available even later on. Going a little bit to Ed, so connecting industry is not always easy. Some research groups in industry, like the structure biology group here, don't really need to be really guided. They know exactly what they want. They know how their problem can be reformulated into a method or a test. They can come to us and do the experiment, and they can go home and do the analysis and make conclusions from the experiments. But these steps are by no means trivial, and they are differently challenging for different types of experiments. So this has been part of our thinking around how to connect to industry, because the needs are so different for different companies. So we have put Tove up here as an expert. We can give her the key to the beamline and she will do the experiment, no worry. And there might be other parts of this company who are not so used, so they might want to have company. And then they can choose what we call mediator company. There are a number of companies, and this is actually a spin-off from Astra Zeneca in Lund. And there's a similar unit in the process, which is present here at SP, who can actually help you doing experiments. A lot of these could be done on the in-house equipment that they might already have in their own laboratories, but they can also support making a visit to the synchrotron to do the final experiment. Magnus Larsson, who sits in the audience, who is our ed equivalent, is starting to build an support function within the MAX4 to support simpler experiments. Of course, you can come as an expert, peer review, which Ed mentioned. But the vast bulk of interaction with companies is that companies come to us through their academic collaborators. A similar facility in the UK, Diamond mentioned that this is up to 80% of industrial visits actually happens as part of industrial collaboration with an academic group. So this needs to be recognized, and we have some nice example where we, for example, met up with ABB, and they had a material they wanted to study, understand most of. We knew what method they should use. We didn't know anything about the material, but we found a group at Chalmers here in Gothenburg who were expert on this material. So we formed a little collaboration to provide that service. So there's so many different models by which you can work with the companies, and we need to keep all doors open because the needs are so different. I think one of the most successful and a sort of an eye-opening experience we had was with the forestry industry. We had Mr. Wallenberg coming in one day, calling up one day and said he wanted to bring his companies to Max 4 because he wanted to understand what the forestry industry, who no longer can sell paper for newspapers, if this could be a research tool that could lift their industry. And we thought that's brave. So we invited experts from SRF and many other places who have been previously done experiments of wood type materials, and this is a multi-scale problem for nanocellulose all the way up to timber. And we do have experiments you could think of doing this. So that led to a workshop with the CEOs of these companies and these experts, and that led to a second meeting where they sent their science directors and people, who eventually led to a joint project, which with some companies being very engaged, and the Wallenberg Wood Science Center in KTH, who actually took the initiative together with the Wallenberg to form this meeting. We have an institute. We also have scientists at KTH and Chalmers involved. And they together formulated both an investment in what they see as another additional beamlines that is required to support this type of research, but also to a national research program to educate a new generation of scientists who can actually use these techniques as part of continued research on wood-based materials. So this is a model we sort of want to adopt as our model. We want to engage industry by industry and make this analysis, what tools and what beamlines, how relevant is Max 4 for life science or for nanoscience or for biomaterials, etc. And what science group are they working with? Are these science groups up to date with what techniques we have, or should we go there and educate them? So this is a model, and we presented it and have the support of the government, actually, one of these groups to do this. And Chalmers at least has really caught on to make this one of the models for them as well. You can learn more about us. We have a website now, finally. This is nothing that's been sort of highly prioritized, but Olivier Baum to sit here done a heroic job trying to get it in shape. We had a previous version, and the first reaction we got to call when people sent an email said, "Have you been hacked?" So it wasn't very good. And now I think we have quite an informative one, and you can read more about different things. These are the people I've shown them that you can talk to. They are here, make use of them. And this is at least part of the stuff that created this facility.